today because the handout that I generated over the weekend matches what I'm asking you to do in the study guide. But that way you'll have something, a piece of paper that you can eventually turn in. But I'm not asking you to turn it in until next week. So if you finish today and you want to turn it in today, otherwise the only thing you must turn in today are some drawings, but they're very, very simple drawings. So I don't think you should have any problem with those. And so you should have time to do all the rest of the things I have on the list for today at some point. So let me share with you the screen of the study guide. Let me make it a little bit bigger first so you can see what I'm talking about. There goes those birds again. I forgot about bringing this up. Before I get to that, I actually edited the study guide. So I made a little bit of a flow chart. I just inserted this. So for blood pressure, I did quite a bit of talking about hypertension. And there is a mastering video that's about 7 minutes long. And they do talk about hypertension and its effects, as well as general blood pressure. So if you just us sending you there, if you feel like you need to see something. And they talk about something which I skipped over and probably shouldn't have, and that's that you can have high blood pressure because you have a high systolic pressure, but you can also have high blood pressure because of a high diastolic pressure. Now, I did mention that diastolic pressure, we were talking about it a little bit today in lecture, has to do with the rigidity and stretchability, but also the sympathetic tone on the tunica media of all the vessels of your body. So as you age, you tend to get less elastic. And so diastolic pressure can rise because of the more rigidity in those muscles. When diastolic pressure rises, you often see systolic following. It will then rise as well. So if you'd like, there is that video. I'm going to point out a couple other videos today too. So for the slide drawings, you're going to be using photographs I took under oil magnification in our lab of our slides. So don't draw this slide, draw our slides. And you're going to be drawing just platelets and red blood cells. So yeah, there are white blood cells on the picture sometimes as well, but we're not going to be worrying about those till after this lab test, when you'll be looking at human blood again and focusing on the white blood cells. So it tells you to draw it at 400 and also draw sickled cell anemia, which I don't think I put a picture in here, at 400. I didn't put a picture of it. So if you go into the lab test 1 materials, you'll see there are, in the histology slide folder, there's a slide of sickle cell anemia and a slide of normal human blood. So I'm having you draw those at lower magnification, not at 1000, so that you can compare the shapes of many of these red blood cells. So we could talk about sickle cell anemia in a little bit, or we can talk about it for a little bit. So maybe we should do that now. Sickle cell anemia is a condition that's genetic, as you may know from introductory biology, depending on whether they covered single gene mutations. This is a single base pair in the DNA that has been mutated, and it's the DNA that's coding for the globulins. So one of the globulins, again, I don't know which one. Now, hemoglobin is normally made, and it has a normal structure, the hemoglobin does. The red blood cells have a normal shape when oxygen levels in the blood are high. But when oxygen levels drop, for whatever reason they drop, it could be because the person is exercising, it could be because there's poor blood flow to some area, that instead of the hemoglobin just dumping off its oxygen, the protein parts of the hemoglobin make temporary bonds to other hemoglobins. So they literally make a chain of hemoglobin molecules that are temporarily bonded together. So what happens is since 97% of the interior is made of hemoglobin, this will often distort the shape of the red blood cells and make them, they're not always sickled, they're not always in a, moon sliver curve. They could have other abnormal shapes. So when you go and look at these slides of the sickled cell blood, it doesn't always look like the half moon. You'll just see abnormal shaped cells, but it does tend to make the cell kind of rigid. So given our discussion today about how blood's going through capillaries, it makes it difficult for these red blood cells to fit through the capillary when a large number of them are sickled. So not every blood cell will sickle, depends on how low the oxygen level is and how severe the condition is at that moment. But what can then happen is some of the blood cells don't make it through capillaries and they clog up and you lose blood flow. And it seems to cause a lot of pain, especially in joints because of this. Now, this is reversible so that when the blood becomes more highly oxygenated, the hemoglobins dissociate from each other and the cell can pop back to its original shape. But given what we're talking about today, remember in the spleen, the bone marrow, and the liver, there are macrophages looking for old and worn out red blood cells. Well, how they tell that they're old and worn out is that their shape becomes, their membranes start to break down and they get abnormal shapes. So whenever a white blood cell macrophage sees this abnormally shaped red blood cell, it's going to remove it from the person's blood. And that's what happens with the sickled cell. They might be doing their job generally, and they might be relatively young cells, but if they're in that sickled shape, the macrophage will remove them. And they'll remove them at such a high rate that it will be impossible for the bone marrow or at times it could be impossible for the bone marrow to replenish the removed cells at the same rate they're being removed. And you end up with a low red blood cell count, which is one way in which you can have anemia. So we will be talking about anemia today in the lab a little bit more because there are various types of anemia. But if you had to define the word anemia, it would mean that you would have low oxygen carrying capacity of your blood. Most often people think of anemia as a low red blood cell number, but there can be other reasons for anemia that don't involve a drop in red blood cell number. Okay, so if there are any questions, let me make sure I see chat as well. Just jump them into chat as we go forward. So those are the two drawings that you're going to finish. One will be looking like this, with normal red blood cells. You can see their biconcave shape. So the two membranes are so close together in the middle here that you can't see the hemoglobin color. And the hemoglobin color in the stain is kind of this grayish, reddish color. So those are the normal red blood cells. And I didn't include a picture of the sickle cell, but you'll see it. So the other part is to work on this handout today. And the handout goes through doing a number of clinical tests and having you write down some of the numbers for these tests. What are their normal ranges? What would be considered normal? It also has you studying, or at least studying the background about plasma. So if we were in person in lab, you would actually be doing this, but not with a full test tube of blood. We'd be using a little tiny centrifuge, a little tiny capillary tube and centrifuging that. And it would divide the blood into its cellular formed element components down here at the bottom, and its acellular part, which is the plasma, the liquid part of the blood above it. So you do that using a centrifuge that sends the heavier stuff down to the bottom of the test tube, or if our case, it would be a capillary tube, and leaving the lighter elements above it. So that's called a hematocrit. And here you're seeing a hematocrit divide into, now these numbers are fine for you to learn, but you actually need to know how men and women differ in their normal ranges of hematocrits. So this is not distinguishing men or women, it's giving something right in the middle of where men and women would fall. Men have higher hematocrits because not only does the bone marrow listen to erythropoietin, but it listens to testosterone. And testosterone tends to mean that in men, the hematocrit is a few percentage points higher than it is in women. So you will be learning the normal ranges for men and women and their differences. So of the formed elements, you had red blood cells and platelets. Do not worry about these numbers. I don't test you on any of these numbers here. The millions and the hundreds of thousands, don't worry about those. You should, however, study over here the components of plasma. This is both, there's no essay on this, but we did cover this in lecture and what are the functions of all these components. So that could, fall into the non-essay based questions on even this exam. So this, again, brings back all the functions of blood. So for the hematocrit, here I have this built in, but this is actually part of that handout. So don't worry about filling it out here. You should know what the normal pH is for plasma. So I think when we talked about it being slightly alkaline. So anything above 7.0000000 is a slight alkalinity. So this is 735 to 745 is the normal range. You will be using this for most of the rest of the semester. You'll need to know the normal pH for blood plasma. Now that's different than the normal pH for intracellular fluid, or for that matter, interstitial fluid, which runs closer to 7.0000. So blood has a slight alkalinity, and we'll get back to this why. So I'm sending you to 2 sites within mastering to do at some point. And one of them, which we'll talk about later, is for understanding a blood typing. The other one is this one, which is a case history. And I'm not going to expect you to come up with the answer for this, but I think it gets you thinking about why testing blood is important. Indicator for lots of the functions of lots of body systems. So to go here, this case I didn't, I think I didn't do it here. I must have done it somewhere because I remember doing it. The little flow chart of how you get there. Now that's the blood typing one. Maybe I didn't do it for this one. But here what you would do is, and I can't type on it right now, you would go to the study guide area. And when you get to the study guide area, there's something, maybe I'll write this and share it so you know how to get there. Let me share that screen. Okay. So you have this in writing. Put that over there. Because I forgot to type this one out. So, and you go into mastering, and from mastering, you go into the study guide area. which is where you're going to go for most things. And then from the study guide area, at the very, very bottom of the menu, you'll see something labeled additional tools. And from that, you're going to go to clinical cases. Once you open clinical cases, you'll see them by their chapter in the book, and you're going to go to chapter 17, and under chapter 17, there are 2 cases, and so you're going to choose from that, fatigued. So the particular case is someone who has a chronic ulcer, and then it's changing his hematocrit, and you'll see some of the outcomes of it. And I think this will help you reinforce some of the ideas of these clinical tests that you're learning the values, the normal values for, and normal ranges of values. So that's why I thought it would be good for you guys to look at it. As I said, I'm not going to go directly on the lab test and test you about this clinical test, case study that they're showing you, but it could be conceptually in there. So, okay, so here is a definition of anemia that is embedded in your study guide, so definitely know this material. There's a number of reasons why anemia can occur. So most commonly, people think of it as a low percentage of red blood cells. So for women who have normal hematocrits of 42 plus or minus 5%, if they had a 32% hematocrit, that would certainly mean they have fewer number of red blood cells and less oxygen carrying capacity. Now, why they might have that is they could, like this man in the case study, the clinical case study you could read about in Mastering, they could be chronically bleeding or even acutely bleeding, which could give them a low red blood cell count. And that would be considered hemorrhagic. That just means bleeding. It could be like our sickle cell patient or a variety of other hemolytic diseases where you cause red blood cells to burst open. That's what that means, hemolytic. Well, here in sickle cell, you are causing them to be taken up by macrophages. It could be dietary. If you have low vitamin B12, vitamin B12 is critical for mitosis. You saw today that red blood cells are being generated at, 2 million per second. So you use a lot of vitamin B in your bone marrow to do B12 to do this. And this is called pernicious anemia. So one thing to be aware of is that vitamin B12 is only available from animal products. So people who are vegetarian don't normally have a problem because eggs are rich in it and they're often eating eggs. But people who are vegan have to make sure that they take supplements in order to get vitamin B12 because it's not available in any non-animal product. And pernicious anemia is a potentially life-threatening condition. So if you're considering veganism, then be aware of that. There are other reasons. It might be that it's not that the red blood cells are being lost, but rather that the bone marrow is unable to generate sufficient rate of red blood cells to replace the damaged ones that are removed. And that's called aplastic. A means without, and I'm not sure how they get the root of plastic here, but it could be damage to the bone marrow due to some drug regimes. Now, chemotherapy is a common one that would cause there to be aplastic anemia. And low levels of EPO, so kidney disease. So if your kidney is not able to produce EPO, you know your liver can, but you may not be producing it at a high enough rate to keep red blood cell production high. So these would affect the hematocrit. However, you can have conditions where you have a normal hematocrit, but instead of being 97% full of hemoglobin, the red blood cells are less than 97%. So hemoglobin is not being made at sufficient rate. This could be a protein deficiency in diet, but it could also be iron deficiency. So we tend to think of anemia often arising from iron deficiency because it's difficult to get enough iron in your diet. That's why iron tablets are prescribed for pregnant women who have not just their own hemoglobin to make at higher rates, but now have an infant that's making hemoglobin as well. There are other genetic conditions like thalassemia, where your hemoglobin is abnormal. And so it's possible that you could have a normal hematocrit, but the red blood cells are carrying too little hemoglobin. So there is a test that, there are two of them actually in your lab manual. One's called a Tallquist test, and the other one uses a device called a hemoglobometer, which looks at the relative color of your blood from the hemoglobin compared to a standard, and then decides whether it's not as red as it ought to be. And if it's not as red as it ought to be, it's possible you have a low concentration of hemoglobin, even if you have a normal hematocrit. Now, these are not necessarily independent. You could have both of these, or having this could lead to having a low hematocrit. So, for example, early in iron deficiency, you might have a low hemoglobin concentration, normal hematocrit, but if your iron deficiency persists, you could end up with both a low hemoglobin concentration and a low hematocrit. So, the reason I'm giving you this background is these are two of the tests that they do in your lab manual. They explain them pretty well in the lab exercise. And I'll show you the handout, or you can look at the handout yourself, and you're going to write down normal ranges, not just for the hematocrit, but for hemoglobin levels. And once again, men have higher hemoglobin levels than women. And one thing I will be a little critical of the lab book is they give the units as milligrams, and it's milligrams per deciliter per 100 mLs. Clearly, you don't have just a few milligrams in all of your blood. It's it's a concentration, so it has to have a volume, and they didn't put that in the lab manual. So again, I have this here, but I don't really give you directions. So down here, it tells you how to get to this blood typing video. It's about 8 minutes long, and I think it will help you understand the ABO blood type and the RH blood type. It's pretty good. There's a few things I'd like to add to it. One is it asks you about compatibility. It does go through that. If you are donating blood to someone else, what would happen if you gave them the wrong blood type? And the difference between the ABO and the RH is alluded to, but not really explained in that circumstance. So I want to spend a few minutes talking about that, and then you can watch this video to really solidify it. I also sometimes turn this, it is compatibility in donating blood, and they do talk a little bit about it being genetically determined, but I also ask you to pull out your old Punnett squares and determine whether, say, a paternity, determine who could be the father. If you have the mother and the baby's blood type, what blood types could the father be? Or vice versa. It could be, if you have a mother and a father, what blood types could the baby be? What I recommend when you do this is use ABO separately and then figure that out with a Punnett square, and then go ahead and add your RH. If you try and do them all, you've got more variables. and the square gets really big, and that can become confusing. So let's spend a few minutes talking about some of the things that aren't in this video that I think are worth talking about. So let me get my iPad going again here, and we'll stop sharing this, and I'm going to share this. There's some good pictures in there which would help you a lot with this. They're not 100% accurate pictures, but there are some pretty good pictures. So one of the things that's not 100% accurate is that you'll get a lot more of in the next midterm is the particular type of reaction that causes blood to clump. So what's happening is on the outside, and they do a pretty good job of this, you have, say you have someone with type A blood. Okay. That means on the outside of their red blood cells, you have a glycoprotein. Remember those guys? Glycoproteins are sugar groups that are on an integral membrane protein. And you have the type called A, let's say. So if this person has A blood type, that means at least one of their parents had to contribute to them the gene to generate this glycoprotein. So one of their parents has to have the gene for that. Once the person is born with type A, and this is a fairly unusual circumstance, their immune system will start making antibodies against type B. Automatically. They don't ever need to be exposed. This is really unusual in your immune system, almost always to generate, well, except for this, to generate an antibody, you have to have been exposed to the antigen. So the word antigen means antibody generating. That's where it came from. So that's how you get antigen. Antigens come in a variety of types, and we're going to do that when we come back and do the immune system. But the particular type that we're going to be working with, and you should know this for the test because we're going to use it, is called IgM. IgM binds more than one antigen. All antibodies do, but IgM can bind 10 antigens. Same antigen, but 10 times. So it's a huge protein, and it's got 10 different binding sites. You can look at a picture of it if you'd like in the book. I'm not going to make an attempt to draw it. Now, in the video, they draw it just as a simple Y molecule like that, and that's not what IgM looks like. It's got 10 different potential binding sites because this is my crude drawing of it, because the A can bind 10 different places. All of these can bind an A. That means that one of these IgM can bridge many red blood cells. So if each of these A's is on a different red blood cell, I mean, this scale is wrong, you can see it can bind quite a lot of red blood cells, plus this has got A's, so you could have another IgM over here also doing the same thing. And what you get is this leads to a clumping. Now this antigen, sorry, I just scrolled it wildly down. If this was A, the antibody here would be called an anti-A antibody. And the person who would make this is somebody with B blood type. They make an anti-A antibody. So if you had somebody with B blood type donate whole blood, meaning plasma and red blood cells, to someone with type A blood, this clumping would occur within their bloodstream, which you can imagine is not a good thing to happen. It could clog up circulation, and they do show and talk about this in the little video. So what I'm adding here to that video is that it's IgM, that it can bind many antigens. You'll have to know this afterwards. And they do a good job otherwise of discussing that somebody with B type, so let's summarize that. If you have somebody with A blood type, they're going to generate anti-B antibodies. And they do this automatically without ever being exposed. So when you're born as a baby and you start having your immune system active, it doesn't have to see the antigen, it's just going to make these antibodies. Someone with B makes anti-A antibodies. And then there is a type of blood, which you're probably aware of, which is O. So O blood means you don't have, if you have type O blood, let me just use the eraser, you don't have any glycoproteins. They're not there at all. So that means that they are going to generate both anti-A and, anti-B. Remember, your immune system is looking for non-self. So if you're O, that means somebody with type A is not you, and somebody with type B is not you. Are we all right? There are, remember, more than one gene. Each time you have a gene, you have two alleles for that gene. In the case of blood typing, some folks have both the A and the B alleles. In that case, their blood type is AB. So if their blood type is AB, A is self, B is self, and they're going to generate no antibodies to either, to any blood type. So understand why this is. If you are type O, that means you don't have any antigens on the outside of your cell, so you can donate that O blood. If it's packed red blood cells without their antibodies, you could donate it to anybody. Now, what I mean by packed red blood is they're going to centrifuge it down, they're going to get rid of the plasma, the antibodies are in the plasma, you can give them that type of blood. Assuming their RH is fine, but if you don't, in a pinch, you could give them O. AB blood can receive blood, whether it's whole blood or not whole blood, from anybody, or packed red blood cells, because they have no antibodies, so they're not going to cause clumping. Now, what I've ignored here is RH. And RH stands for rhesus factor because it was discovered in rhesus monkeys. Rhesus factor is also on the red blood cell. My red blood cell's getting a bit crowded here, but let's put some rhesus factor on there. If it has the R8 rhesus factor, which is sometimes called D, I don't know why it happened to C in all this, and let's say they have their type A blood again, then this person is going to be considered A positive blood. So if it's present, you add a positive to the blood typing. Greater than 95% of the population of all races is Rh positive. So the vast majority of people are Rh positive. And that's why I said in a pinch, if you don't even know the Rh, you can give O blood, packed red blood cells, to anybody. Let's say this person is Rh positive and you're giving it to someone who is Rh negative. That could be a problem. But what it takes is, in order for the person to make antibodies against that Rh factor, they have to be exposed to it previously. So this requires, let's start here. An Rh negative person requires previous exposure. And the video talks about this, to an Rh positive in order to make antibodies. Which would be anti-D antibodies, anti-D, you could also call them anti-Rh antibodies. So let's say you had an accident victim, they're bleeding, and you wanted to give them packed red blood cells to help ease their low red blood cell number. Then you could risk giving an O positive person's blood to any of these other blood types, hoping that they would never have been exposed to the Rh factor. But in an ideal world, the really true universal donor would be O negative blood. That way, there's nothing on it, because negative means you don't have the Rh factor on, and there's nothing to react to. So this is really the true universal donor. The most rare blood type, and you should study the little table in the lab manual, is AB negative blood. As I mentioned, more than 95% of the population is Rh positive. AB is low. I don't remember the percentages. You don't have to know the numbers in the table, just who's the most common, who's the least common for the different races, and the races are all the same. So you don't have to worry about that too much. So the other place this comes up, and the video alludes to it and the book talks about it, is in pregnancy. So if an infant is not born yet, is conceived, who is Rh positive, to a mother who is Rh negative, if this is her first child, then there's usually not a problem. But in any pregnancy, near the end of pregnancy, the placenta begins to break down and the mother and the baby's blood mixes. So the mother becomes exposed to the Rh factor in the baby's, her first child's blood, and starts to generate antibodies against that baby's blood. Normally, though, that first child's born before the mother has a chance to make a sufficient number of antibodies to cause a problem for the baby. However, if the same mother gets pregnant a second time with the same father and has another Rh negative child, then, or conceives another Rh negative child, her immune system is much faster and much stronger against the Rh. So again, near the end of pregnancy, the placenta breaks down, which it normally does. Mom and baby's blood mixes, and now mom very rapidly, within a few days, starts to generate large numbers of antibodies against the baby's blood. And this can lead to the baby being born quite anemic. They used to be called blue babies because, their normal pinkish skin, or gums are not the normal color, and they're quite anemic. So nowadays they have drugs to suppress the mom from making the antibodies, and you don't see it as a big, huge problem. Plus, it's fairly rare. But if you end up being a woman and you're Rh negative, you should be aware of that, because if you become pregnant, that has its consequences. Okay, any questions on this part? Okay, so let's go back to the study guide again. And I'm just leading you through that. So there's where you go to see this video about blood typing. It's more or less accurate. It's just some of the things are a little unusual, like the antibody only having 2 binding sites, not being an IgM antibody. And they don't fully explain the RH. They say it, but they don't really explain that antibodies have to be triggered in most cases. And the RH is one of them. So I think that's the end of this study guide. Let me leave this. I guess I have to move you guys is the problem. Okay. And you'll see that over the weekend, I have put into your folder here this clinical assay handout. And what I started with, because we can't do labs in person, but I think this is one of the critical things to get out of this lab. I continue to use real human blood where you are testing your own blood, which is obviously a huge biohazard. They'd probably have a heart attack if they realized we're doing this. I do it in a very paranoid way so that I make sure you only handle your own blood and that you never handle any of the equipment. So this lab manual exercise starts with a series of rules about handling blood, human blood, but it could be any other potentially contagious human secretion. And what I did is I wrote this about page-long, maybe entertaining story about a lab technician who isn't always following the rules. So I ask you to read through this. You can highlight if you want, or you can just list afterwards. I leave some space for you to list at least 10 mistakes. And ideally, I mean, there's sometimes he makes this, or she makes them, I guess it's a he, makes the mistakes more than once, the same mistake more than once. That's okay, you can pick them out, but I could easily find more than 10. So if you can either list, or if you want, you can just highlight or number and send this back to me, so that you come away from this lab at least with an inkling of how to go about properly handling potentially contagious body secretions, including blood. And then within the lab manual, you can find all of these numbers. Make sure it's a range. So you're giving from, this percent to that percent, or, an average of this plus or minus that, so that either one. And then I didn't leave you much room, but I gave you answers for this in the slides previously, why you could have a low hematocrit with a normal hemoglobin concentration, or a low hemoglobin, and you could have a normal hematocrit at the same time. If you've watched the video, you'll understand how to determine what the blood types are in these folks. So we use a little card to tell them on, not little wells like in the video. There could be pictures of those cards showing you clumping or no clumping. And what does that mean? You have to interpret it, including anti-D, remember, is RH. And then I already answered this, but who's the universal donor, who's the universal recipient, that kind of thing. And which are the rare, which are the common blood types. And then I just explained this, why an RH negative mother can have a successful first pregnancy, but has the baby at risk if it's an RH positive baby in a second pregnancy. And then finally, coming up with blood types. So I give you a mom and a dad here, and then I ask you to list all the possible blood types of their children. I suggest you play around with this. Sometimes I give mom and baby and ask for the possible blood types of the father. And try this one, and if you're really struggling with it, we can talk about it in the review lab later. So lots to do there, and I'm going to turn you loose for whatever it is the next 40 minutes or so, 45 minutes, and you can work on the slide drawing shouldn't take you too long. So remember, you're going into the histology slides and you're going to be drawing a normal red blood cells at 400 and at sickle cell red blood cell. And I said draw them at 1,000, but that was what the picture was taken at. You're not going to do them at 1,000. You're going to do the 400 ones and see the sickle cells. At least I believe they're at 400. Let me just change this. And I have to get up to the top. I believe if we go in the histology, I believe the normal blood, I guess it is at a 400 here, and sickle cell at 400. So I do have some at 1,000. Maybe I did have you draw platelets maybe at that because they're so tiny, because you're supposed to draw platelets and red blood cells. So I would draw this one and then draw, so 3 drawings to submit today, these last 3 things. But they should be pretty quick drawings. You don't have to worry about the white blood cell on there. And submit them by 5 o'clock today through the assignments. And then the handout of the blood isn't due until next week. So you'll see that's been inserted in your assignments too as another percentage. And I'm trying to remember, is your blood pressure due today? No, I think it's due Thursday, right? Yeah, it's not due till Thursday. So this Thursday, you're turning in that blood pressure handout as well. Okay, so I'm going to be here if you need me. Let me turn on chat so you can always send me a chat. If you need anything, just call out and I'm happy to answer questions. If for some reason or for whatever reason you would like to be put into a breakout room, I can do that as well. So just leave that in chat and I'll put you into a breakout room if you want to work with other students on any of this.
Professor, for that blood pressure assignment, it's just the blood pressure assignment that's due Thursday, right? The blood clinical or the clinical assay is due on the 29th?
That's right. Yes.
Okay, just checking because it says due September 24th, you put blood pressure results and blood clinical assay.
Yeah, I was waffling about it because I thought, oh, you could do that too. But so I probably didn't change the title, but I think the contents, when you open that assignment, it says just the blood pressure results. So sorry about that.
It's okay. Thank you.
Mm-hmm. Okay, any other questions? Okay, folks, give me a shout if you need anything.